You're listening to the FaceTime with Scientists podcast, where we go into the lab or into the field to learn from scientists about their lives, their work, their struggles, and their joy. Let's go. Today, we're here with Nuri Smith at Emory University, and I'm super excited to chat with you. I love your Instagram account and all the stuff, and TikTok, all the videos that you put up about your organoids and assembloids, which I have a question about. I don't actually know what assembloids are, but in any case, super excited to chat with you today about your research on ALS. So, yeah.
Awesome. Well, I'm happy to be here as well. Yeah, I'm excited to tell you about what we do, like in our lab, but also broader in terms of the organoid space, because I think there are some exciting things happening in the field right now. So I am a rising fifth year at Emory University. I'm getting my PhD in genetics and molecular biology. Fifth year sounds disgusting because I let the time went by so fast. I'm a rising fifth year and I'm a part of Jimena Anderson's lab. And so she's a newer PI and she did her postdoc under Sergio Pasca. So like really big name in the organoid field. And a lot of her postdoc work was in establishing this Assembloid model, which we can talk a little bit more about. And so now we kind of take that model along with spinal cord organoids and sometimes even cortical organoids to understand ALS as a disease. So this disease where there's this loss of motor neurons, a big part of my project is using this model to, first of all, we have these motor neurons and we have them derived from ALS patients and healthy individuals. And so I can ask questions like, how are these motor neurons degenerating? Because they degenerate in our model, which is really cool to see. And what I'm working on right now is profiling them at a transcriptomic level. So looking at differences in gene expression and seeing at that level how these motor neurons are different and some other cell types.
That's super cool.
Yeah.
So, a few years ago, I worked for NIH, the Center for Alzheimer's and Related Dementias. So, there, I feel like maybe there was some similar things in terms of like they were really trying to figure out molecular mechanisms of dementia and looking at it from like a genetic standpoint, as well as like looking at individual cell types throughout the brain, like what was going wrong in those cell types, and then building kind of models of, well, you need to have different cells together, you know, to kind of recapitulate the brain is so complicated that you need to have multiple cell types and be interacting to really be able to tell what's going wrong if you introduce a mutation or something. So I learned a lot about that. But yeah, how is ALS different? I don't know a whole lot about ALS, but like, is it related to dementia? Other
Yeah,
types of
so
dementias?
it falls into neurodegenerative diseases. So all of these diseases where some cell type in the central nervous system is degenerating. So when I was in my postbac, I was doing Alzheimer's research just a little bit. I was mostly working on schizophrenia. But yeah, it falls into that kind of umbrella of neurodegenerative diseases, with each disease usually having a certain cell type that degenerates more than the other. For ALS, it happens to be motor neurons, but you think about like Parkinson's, which is part of the dementias, it's usually these dopaminergic neurons that are degenerating.
Gotcha. And then have you always been interested in ALS? Like, how did you?
I haven't actually.
Yeah.
So what attracted me to this lab was the model, and then I fell in love with ALS afterwards. I've always been in kind of like the neuropsych space. I love like psychiatric disorders and understanding them from a molecular level. And I think I'll probably return after my PhD to more of that work, but certainly like I've grown to love studying this disease and then also just getting to see some patients from time to time and like hearing their stories just makes the work more fulfilling and like gives me more purpose, I guess, if that makes
Yeah.
sense.
Yeah,
Yeah.
totally. Yeah, that's amazing. Yeah, when I worked at Card, it was like, yeah, a lot of people would talk about stories about telling the public about their work, and it was like just people had such a reaction to like, I'm so glad you're studying that, right? Like I know someone with dementia, my grandpa, my grandma, right? And so like it makes a big difference to people that someone's working on this.
100%. Like I didn't know much about this disease outside of like the ice bucket challenge when I was in high school. And now that I study and I people ask me what I do and I tell them like everyone, a lot of people have a story or they have like even family members that are distant, they have a story. So it's been humbling and also like rewarding to be able to contribute to this field in a way.
Yeah, that's amazing. Well, okay, can you tell us a little bit about organoids? Because, yeah, when did they come about as a thing? Like, I feel like I only learned about them recently working at CARD, and yeah, like when I was coming up as a scientist, I very much studied like kind of, cells on a dish on a flat surface, but that like the fact that our cells don't normally work like that is important to, yeah, how we study them and how we look at what's going wrong. We need them in kind of this like three-dimensional system.
So organoids came about, I want to say like 20 years ago, but not as that name. Um, what was happening, well, scientists were making this observation when they looked at cells in a dish. Sometimes they would form these spheres by themselves and they called them embroid bodies.
Oh.
And then when they looked at them to see what they were a lot of like neurons, usually like balls of neurons, but then they would start differentiating on their own to be like certain types. And so I think over time, people have just built on this idea of the embroid body and use certain patterning molecules to push it a certain way. So when you're pluripotent and you're like this ball of cells, you have certain patterns and factors that are acting on this ball of cells to pattern it over time, right? And so what we do is take advantage of those molecules and we do that in culture. So we can take these pluripotent cells and you can make cells pluripotent via different ways. But for us, it's like you can take skin samples or blood samples, and then you can give them what's called Yamanaka factors to push them back. So these faded cells get pushed back to being pluripotent. And then we use these other factors to drive their differentiation. And so now there are so many different types of organoids that exist. But for us, we care about the central nervous system. So there's different brain regions that have the cell types that belong to that brain region. And then for my research, it's a lot of the spinal cord organoids.
In our brain, like in a whole organism, it's so complicated. All the neurons are communicating with each other, or glia cells are supporting it. So it's much more alike to what's really going to happen in real life if we study them like kind of in this combination. And it's like,
Mhm.
can you maybe talk a little bit about like how that's kind of how it's changing how we study neuroscience now that we like have some of this, we're able to study this in a more complex system without like maybe going to the animal model right away.
Yeah, I think you've like touched on a lot of like all of the important pieces already, which is that for a long time it was animal models, and then you have either these single cell cultures or you might co-culture with one other cell type.
Hmm.
Obviously, we have so many different cell types that are working with one another, supporting each other, and making a healthy, central nervous system. Like neurons themselves just have so many different types, but then you add in the glia, which are supporting it. Well, actually, I just attended a talk that said we shouldn't refer to glia as support cells anymore because they do their own thing.
True,
Um,
yes. Yeah, I feel like, yeah, that maybe it's an older idea that they just were there to support, and now we're finding like they do a lot of other things, right? But
yeah, like they're the rock stars of the central nervous system. Like we have to be honest. But yeah, so like with the organoid model, we have a lot of these cell types that we see in human models. I study humans, so I think that's what I care about the most. But like most of those cell types we have in our organoids, and if we don't have them, we can do a process called engraftment to put them there. So like our spinal cord organoids and cortical organoids, they don't have microglia, but we can grow microglia in 2D and put them in. And all of this is important because we're getting closer and closer to trying to model the human brain and spinal cord. And that's important because a lot of the diseases, especially when we think about neurodegenerative diseases that we already talked about, there aren't spectacular animal models for these. We have to kind of induce a human disease in them, especially in ALS. Like
right.
you have to give an animal ALS versus studying it in this kind of native background that the human had that we're studying um, these organoids with.
And then you referred to, and you also sent me a study on assembloids. Are those different?
Yeah, assemblers are so cool. They're like one of my favorite things ever. So assembloids are basically when we take these already patterned regions, so cortical, spinal, or some other version, and then for us, it's with muscle as well. But assembloids just means you're putting together different regions and they tend to fuse together on their own and they communicate with each other. Cells will migrate, cells will connect. And I'm going to show you some of our assembloids where we have spinal cord and muscle, and that muscle will actually contract and signal as well.
Wow,
Yeah,
that's
it's actually kind of
crazy.
freaky.
That's crazy. So it's, I mean, you're almost starting to create a little bit of a Frankenstein, almost a little bit of an organism, like, on its own.
Yeah, it's kind of freaky, like, a lot of people that I talk to about it, they're like, "Wait, like, are these, like, alive?" And I'm like, "No, like, there's a lot that goes into being alive and being conscious." But yeah, like, the cells are functioning in a way that you would expect, which is kinda crazy to see in a dish.
Yeah, that's
Mhm.
super cool. Well, yeah, can you maybe talk a little bit, kinda like dialing back of, like, why your lab studies organized, like, what are you doing with these things in terms of, like, experiments or looking at how ALS works? Like, how does yeah, studying organoids and what are you interested in particular? What does your research entail in terms of ALS?
Yeah, so our lab and I guess when you first asked the question, my brain went to, like, our group, and our group is, like, quite big because we have, like, 3 labs kind of all using organoids and we all use them for
Uh,
different things. So we have some people that are like doing autism work and some people that are working on specific genes like CACNA1A. Our lab is very focused on ALS, like I said, and within that we have one person that is very interested in neuromuscular junctions and but her first paper that she's about to put into preprint very soon is just going to be about establishing this model. So we specifically have focused on a mutation in ALS called C9orf72. And this is the most common ALS mutation. So like 90% of ALS cases actually are what we call sporadic. So there's no known genetic link. But for the
right.
ones that we do know have a genetic link, the most common one or genetic mutation in that is C9orf72. And so the background for a lot of our ALS lines and a lot of our research is in this line. And so her work is about establishing this model. What phenotypes do we see? How alike are they to patients? Which is really cool. And I won't spoil her paper before it comes out, but like there's a lot of cool phenotypes that we see. And then my other colleague works specifically on oligodendrocytes, so these cells that wrap around the neurons and allow it to conduct signal. And they also give what's called neurotrophic factors, so support. So they're also another glial cell, but she's interested in the biology of those cells. And for me, I already mentioned I'm interested in the motor neurons, but a lot of my work right now is profiling all of the neurons in this disease from a transcriptomic level and
Mm,
then also moving into more functional things now is where I'm kind of focusing my PhD work.
that's super cool. Well, random aside, I remember when I learned about the oligos and dendrocytes and you kinda like, because it's like the neuron, right? You think of it's like the cell body and it has this long, like, axon, a long
Mhm.
extension to, like, reach to another neuron, and then this like, I feel like in textbooks, it always shows this, like, myelin sheath, right, that's, like, covering on the axon. It always
Just
just
like by
shows it as,
itself.
like, by itself, so it's like,
Uh-huh.
okay. I mean, I spent most of my life as a scientist and a biologist just thinking, like, these are just little, like, I don't know, protein coverings or something on until finally one day there was like it showed like, "Oh, no, that's another cell that's, like, holding it basically." And I was
Mhm.
like, "Why did I not know this?" And so, like, even that idea of, like, how we picture science is how we show it, like, if we show it only on a 2D surface of a cell or something, like, we're getting not that full picture. It was wild when I realized that that was actually coming from another type of brain cell.
No, felt like it was wild for me too. And I think most of the time when you see it in textbooks, it's showing you like peripheral nerves where it's like a Schwann cell only doing the wrapping. So you just see like the myelin. But like in the central nervous system, yeah, it's these oligodendrocytes and they're cool cells I've learned since paying attention to her work. But yeah,
Yeah.
I think that goes into, like, how we show science and, like, show it as art, which is so hard to do, like, it's so hard to do. I used to be interested in that as well. Yeah, my brief aside.
No, that's amazing. Well, not to, like, take us too far aside, but might as well. I mean, your Instagram, you've grown a large following for people where you show your like, under the microscope, and you show what you're doing in the lab, and, like, how is that yeah, when did that start, and how has that changed or influenced how you do science?
You mean, like, my social media?
Yeah,
Oh,
just in
um,
your science communication in general.
um, uh, how do I say? So, I am a first gen college student. And certainly the first to pursue science in my family. And so I struggled a lot when I decided to become a scientist, but also like I was pre-med before because I thought I wanted to be a doctor. I did not. But like I struggled in my classes a lot. And when I realized I wanted to be a scientist, I didn't know what that meant, like at all. Like I had no concept of a scientist outside of like astrophysicists like Bill Nye. Like I didn't know like about biology as a field. And so I struggled a lot with concepts and learning and all of those things. Um and I used to just be so like frustrated that there were like no resources for like no entry point for me into science. So that's anyways, that's my beginning. But as I've gone through my education, and especially now with the current administration, it's very evident to me, but also other scientists, that we have to be responsible for making sure people are understanding science because it's my personal belief that scientists are public servants. Like we do science and we're pipetting and doing all the things, but we do have to let people know what it is that we're doing. And so I think that's really important to me. And through going through like so many meetings in my program, my PhD program, I mean, um, there's always been like this frustration from other scientists and professors about like people not understanding science or like not getting
Right.
it and, those things. And I had my own frustrations about um us just not doing enough for people to understand science, I guess. And that's kind of where my social media was birthed. Like, I was like, I cannot expect professors to do something I won't do. And so I was like, okay, I'll try to talk about science because it excites me and I think it's awesome. But I also want to do it in a way that people can understand. And I think I'm working on that every day,
Yeah.
honestly. That
No,
was a
I mean,
roundabout
it's
answer.
no, it's so true, and it's so important, you mentioned briefly, right, that you take and I know at NIH card, they did the same thing of, like, a lot of these organoids are actually grown from, like, a skin cell or something from an actual patient where in that way, I guess you're getting some of the maybe genetic mutations or some of the, biology from that might have been there in that disease originally, and then forming the cells that you need to look at that biology in a neuron, but just that idea of, like, we're taking cells or samples or even brain samples from patients, like, that have donated their bodies to science, like, there's a there's a responsibility there of, like, being good stewards of that and then also sharing that back with the people that have participated in science, like, not just keeping it here until we publish for other scientists, but, like, spreading that back out to the people who need it, yeah.
Exactly. Like, that's just disease research, but there's so many people that study basic sciences and nobody knows what it is that they do. And sometimes people don't care because it's like, oh, you're not studying a disease. So I think we don't get the skills as PhD researchers to communicate science to other people, but I think that we should. Because if people understand what you do, then they'll vote for it and they'll, you know, vote for our funding and things like that. So I think it's so important that we share what we do.
Yeah. Oh, that's amazing. Well, did you want to, like, show us any stuff in the lab, like, so we
Yeah,
can, like, yeah, see it and chat about it?
I would love to. Okay, so we as I mentioned, we're like three groups. So there's my PI, um, Hamin Anderson, Fikri Bire, and Stephen Sloan. And what they did was they because they all came from Sergio Pasca's lab, they wanted to kind of standardize organoids a little bit better and create some methods, I guess, of what is the word I'm looking for? Standardization, basically. So what they created was the Brain Organoid Hub, and I'm taking you there
Hmm.
now. So a lot of our work, like I don't have to form my own organoids because we have this Brain Organoid Hub, and it's really awesome. And one of our hub workers is actually working. Okay, so this is the hub space. She's working. It's just like our codes and all of our organelles live here in these incubators, which is really cool. And I'll show you one more thing because I think it's awesome. So all our organoids are formed there, all of our cortical, spinal, and all of the other regions. So it's super helpful for me as a PhD student to not have to form and like feed all of my organoids all the time. Um, but we just recently got a really cool robot that forms our organoids, feeds them, and even takes pictures of them as they develop over time, which is really cool.
I think they had something similar at Card that was very much like a fully customized, like, to do the things that they needed to do. So it's yeah, it really and like you said, this idea of a hub where you don't have to, like, always grow your own, like, I'm sure it just makes the science so much like you said, you can standardize, you can compare your experiments on organoids to other people's organoids because they're made in the same way, but then just being able to, I'm sure, do the science more quickly because you're not having
Yes.
to create those by yourself.
Exactly. And the like I said, the initiative was standardization. So all of our protocols and everything that we do is available online for people to use, all of our recipes, which is really cool. We came at a good time. So this is the robot. It is currently feeding. I don't know if you can see that in there.
Oh, yeah, I can.
Yeah. So in those 96 well plates, that clear plate right there are organoids. And so it's feeding each organoid that is living in there. So each well has an organoid in it. But it's a really cool system. And I think it's showing us, yeah, it's currently showing us the organoid. So,
Oh, wow.
so this is an organoid. I should think somebody's remoting in.
That's kinda awesome.
Yeah.
Yeah, because 'cause they're, like, looking at it specifically. So I guess it's so dense in the middle, it's seeing black because it just can't
Yeah,
see through all of those
exactly.
cells.
So the view that you just saw, so this view is like all a lot of wells at once, and each one has an organoid in it. But yeah, some of them definitely remoting in, but that's helpful for us in our demonstration purposes.
Yeah, that's amazing. That's so cool that you can be, like, at home and be like, "How are my cells doing? How are my organoids doing?"
Yeah, it's really cool. I haven't used this yet. I will have some being formed in here soon. I'm just scared to touch it. Like it's like $1 million. I don't want to touch it.
Yeah. Yeah.
But yeah, it's really cool. Um, I have other things to show you. With are the assemblies that we were talking about. Um, but they're really cool. So I have two to show you. One is calcium imaging, so we can actually look at the individual muscle fibers and their calcium signaling, which is really cool. So you'll see like little bursts around. And then the other is, like I said, the muscle actually like being connected to the spinal cord and like, ugh, it's kind of nasty to me, but like you'll see it kind of undulating and like firing a little bit.
That is crazy. Yeah.
Yeah.
How long
I
have you been working with organoids now? Like, have
since I
with these
started
systems?
here, so almost, I mean, technically academically, I'm a fifth year, but it's 4 years.
That's wild. I feel like it's such a, well, and I feel like now it's becoming such a useful skill, like, if you've worked with organoids, like, it's a maybe a very in demand skill for, like, lab researchers to know how to create organoids and to deal with them for, like, studying disease.
It is a skill and that like is a bit in demand because I think for a lot of grants now, some of them are requiring that you don't use animal models or you
Right.
add like an organoid or an iPSC derived model as well. Okay. Okay, I'm gonna put this under the scope, but those like opaque regions, those are the spinal cord organoids, and the clear region it are muscle.
Gotcha.
And so we grow them on what's called a transwell. So it's like this little mesh that they're on sitting on top of, and their food or media is on the bottom.
Gotcha.
Yeah. All right, let's put this on the scope. Hopefully it behaves for us.
That's interesting. Why do you grow it, like, above the media?
Just because they will grow together
Ah,
better. Yeah, like we put them together and then they'll fuse. But you could
gotcha.
ew, okay, it happened. Sorry. All right, let me turn the light off. Okay, so you can see it. It's in green. Can you see that pulsating? 1 second, it'll do it again. There it goes.
Oh, yeah, I think so.
This is like spontaneous. I'm not making it happen, so we just have to get lucky. Hmm. Oh, now they want to be calm.
Yeah, it's the observer effect.
I'm telling you,
Even the color of it just being like this green glowing, like
a green orb.
So, yeah, is it something where, like, you can measure or, evaluate, like, the level or the amount of that pulsing, the activation of the muscles or the amount
Yes.
of it under different conditions? Is that kinda what you're looking at?
You can do a few types of experiments. One, or at least one way that I've seen the data collected is like you can measure the strength of that kind of contraction.
I
You can measure the
got
strength of that contraction um, in a few different ways. Or we do some other really cool experiments where we can put rabies on this muscle and we can track the cells that are connected to it. So rabies travels retrograde. So like if you were to get bit by some rabid animal, that virus travels back to your brain. Basically, it jumps synapses back to your brain. And so we can harness that in the lab in a safe way to figure out what cells are going to be connected to our muscle. So like we can count those cells and in the context of ALS, we can see if the ALS ones have less motor neurons connected to the muscle, things like that.
That is wild.
Yeah. I probably definitely will make a video about that soon, about rabies.
That is crazy.
Yeah, viruses are cool in the lab. Like you can use them for so much stuff.
So you're just using that to, like yeah, that's a really interesting thing to me. I talked to some folks at LSU one time about, like yeah, it's not necessarily easy to study even in neurons, like, which ones are, uh, connecting to which other ones and actually communicating. So,
Exactly.
yeah, the fact that you can use the virus to, like, to tell which ones are actually connecting to the muscle is super cool. And are you actually were you was that, like, looking at what specific cell types were? Or yeah, like, what did that tell you, I guess?
Yeah, you can. So for me, it was just to figure out if those cells were motor neurons and count how many there were. But
Gotcha.
there are different ways of doing this. So another grad student used this rabies to figure out what glial cells were connected to the neurons. And I thought that was really cool. But like the implications of rabies for research are wide. But yeah, there's a lot of ways you could do it
That's so
or
cool.
use it.
Yeah.
They don't want to behave today, but it's okay. I can show you other things. That's just the way. Like that's just how science goes,
Yeah.
sadly.
Yeah.
All right. I'm going to take you to our scope room then. Speaking of viruses, we use a virus that lets us look at calcium signaling in our organoids. So this is our scope room. This is our confocal microscope. I'm just going to use fluorescence today, though, not the actual confocal pieces or software. Um, that's our plate. another transwell, but these organoids have the virus that allows us to do calcium imaging on them. And we'll visualize here on our computer screen.
That's super
It's,
cool.
it's it,
Yes. What is the why study calcium signaling, I guess?
oh, great question. So calcium signaling is one thing that has been shown to be, it depends on the paper you look at and the stage, but in ALS, calcium signaling can be really high or can be really low, depending on how much motor neuron death there's been. But motor neurons typically suffer from what's called excitotoxicity or hyperexcitability, so they fire a lot. And we're using these assembloids kind of as a model to read out that activity. So if they're firing a lot, maybe the motor neurons are causing this muscle to fire a lot, basically. So you can measure or see that signaling here in the muscle.
What's funny is that, like, I was just talking to actually an ant researcher at the last podcast, but she's studying the ant colony in a way, like there's aspects where it operates a little bit like a brain. And she was talking about there's this aspect of like social contagion that you could study where like people in a crowd, maybe they all look in one direction or like there's a loud noise or some, basically people start to panic and it'll become like this self-reinforcing action where it's like just like a mass panic and maybe it causes more harm than good. And she was saying social insects like ants, sometimes they're actually very much immune to this. And it's almost, she was comparing it to like synapse pruning or like how in the brain there's actually aspects where like you need some neurons to kind of quiet other ones so you don't
Yeah.
have this hyper excitability that turns into dysfunction,
Hmm.
which I thought was, like, really cool to think about the ant colony kinda, like, right, going crazy and not, kind of, making sure that each other is like, "I got this," kind of thing. That, like, how does that, compare in disease? Sometimes you're having actually maybe too much firing between neurons that creates this, like, yeah, dysfunction, and you're thinking about guess on neurodegeneration, I would think about neurons dying or becoming, like, quieter when actually maybe the pathology is, like, too much action.
Mhm. And I mean, one of the what you brought up about the ants, like some function to like quiet things down, is also a theory. And I mean, there's some evidence in ALS that like these inhibitory neurons that are going to function to quiet things down are actually degenerating themselves. So they can't put the brakes on the
Mhm.
signal. And so the motor neurons are just firing even more. So there's a lot of, like, speculation about what's going on. I have my own theories that I'm investigating. But, yeah, like, I didn't think ant colonies work like that, so that's really cool
Yeah.
to even think about. That was a cool connection. Alright, I see a little bit of twinkling. We're gonna have to focus and look, um, and then they stop. That's so funny. Okay, can you see this?
Yes.
Well, okay, we're gonna just look for a second because they tend to be quiet at times and then sometimes they're really active. But if you just saw that in this, you can see the signaling ever so slightly. Can you see this?
I think yeah, I did see kind of yeah, I saw it kinda, like, disappear just now, and then it kinda came back a little bit.
This is so funny that today of all days, things decide to not work how they usually do. That's so just fired.
I'm just, like, waiting for, like, fireworks.
No, they're not like super like, I mean,
Yeah.
they can be. We have to stimulate them for that, and we don't typically stimulate them, but they can be. Let's see if I can find a more active ones.
I guess what experiments would you use to compare maybe activity of the calcium signaling? Would you have different organoids or different ones that are, like, have different genetic mutations in them or
Yes. Typically, it's like a comparison between the ALS and, like, their healthy sex and
Gotcha.
age match control is usually the comparison that we're making, but we also have what we call isogenic lines, and so those are the lines that we just remove the C9 mutation. And we can look at difference in calcium signaling between those two as well. But this one from there,
Oh, yes.
you can see a lot better. Yeah, this one
Yes.
is she's twinkling. And then it stops for a while.
That's crazy. Oh, yeah, I definitely see it now.
Yay!
Yay!
She can
That's so
see.
cool. So
Yeah.
so when what we're seeing twinkling, is that individual
Those are
neurons?
individual muscle fibers.
Gotcha.
Yeah.
Right, right, gotcha. That would be, yeah, muscle fibers that are twitching. And so
That
the
are
muscle fibers themselves, they have viruses in them that you're using for the labeling?
yes. So the muscle fibers have a virus that has what we call GCAMPs. So these are calcium sensors. And so here is the spinal cord organelle. And when these fuse, you can't see the motor neurons individually here unless we label them, and they're not labeled, but these are going to connect to a muscle fiber. And so as this motor neuron is kind of communicating with this muscle, you'll see this calcium signaling that actually is green, but it's just showing white here. So it's like glowing green. Um, yeah, as it's firing or as
Wow.
it's contracting. But yeah,
Yeah, that's
that's
so
how it
cool
looks.
to be able to build
Mm.
this, like, just from individual
Ooh,
cells being wow, that was, like, a
sorry.
big one.
Sorry. But yeah, what
Uh,
were you saying?
that it's so cool that you can, like, build yeah, just from individual cells, like, start building this kind of functional system that would be I guess, right, in this case, the muscle fibers aren't doing anything. I mean, other than pulsing they're not, like, moving an arm, but,
Yeah,
like
exactly.
but in a disease case, what would all that extra firing do? Would it make the muscle weak or twitch or
Yeah, thank you for asking this question because I didn't explain it in my frenzy of trying to like set things up. Um, so in ALS patients, some of the earlier symptoms are spasticity and like stiffness of the muscle. So like people have these muscle spasms and then they can like again have this stiffening. So having that as like a early symptom, maybe we can show that in our organoids is yet to be really proved. But like it's one thing that we can kind of use to see just how good our model is in modeling ALS.
right. That's super cool. In looking at these models, is the goal or some of what you're doing, is it on the diagnostic side? Is it on the therapy side? Or is it just understanding the disease?
Just understanding for now. I think the goal eventually is to like test some of these therapeutics. Like we've tested some drugs in our organoids and it's kind of like mixed, but also it's mixed in patients, which is really interesting because like a lot of these that have been approved were in animal models and testing them in our organoids, we kind of see what we saw in clinical trials almost. Like it's a mix of whether they work or not. So I think that's another way that organoids are cool because of the genetic background that they come from. Maybe they're another great model to study treatments for the disease and therapeutics.
Yeah,
But right
I
now, we're
just
just trying to understand.
right. Like, that just brought to my mind I mean, I guess you could imagine, like, an individual patient with ALS, like, creating organoids from their cells where you could test almost like a precision therapeutic, like, for them.
Exactly that. Like organoids are, for a lot of people, where medicine might be going, like this idea of personalized medicine for certain, diseases or disorders. Um, yeah, I think they're a cool model for that for sure.
Yeah, that's wild. That's so
Seems
cool.
like
What
science fiction.
it really does. It really does. I mean, yeah, what's your, I guess, daily experience? I'm sure it gets, like, old hat, but what is your daily experience in the lab, and, like, maybe what do you find really cool and exciting? And then maybe on the other side of that, like, what's difficult? And you talked about some of that, I think, with the current administration, we have no dearth of frustrations in science right now, but maybe
Yeah.
for you personally.
My daily experience is actually pretty awesome. I cannot complain. Like, outside of just PhD stress, which is like, when am I gonna graduate? I have to produce all of this data. I will say that I'm very fortunate in that I have amazing support and great mentors. And so the PhD is never hard in aspects of like other ways that I've heard where people have bad relationships or like difficult relationships. I think my day to day is good, and I've made like a lot of friends in my PhD and in my lab. So I have a lot of fun doing my research. Also, I'm just, being frustrated with figuring out my next thing, which is just a part of the work. What I really like is I like mentoring and teaching younger students. That's probably like one of my favorite things. I like to explain science to people, as you know. But I live for like their like aha moments of like, whoa, like this thing that I learned in class, oh, this has an application and like I can see it and I can like touch it with my hands and I can do stuff with it. That is so fun to me. Um, and as far as what I dislike, I think I don't know. Like, I don't think dislike anything where I'm at right now. It's more like worldwide, I guess. Like we did talk about like the administration. Um, but I think it's also kind of old mindsets in science I dislike, which is like we are kind of like put up in like these ivory towers sometimes. And like I said, we don't come down and communicate and talk about science. I strongly dislike that. Like I hate it actually.
Yeah,
Um, and so I think I don't know, I just have this strong drive to like leave science better than I found it and help, I guess, the next generation of scientists, even though I guess I am the next generation of scientists, like make it better and like make it more inclusive and make it more accessible for other people. Yeah.
I
Hope that
love
answered your
that.
question.
Yeah, leaving it better than you found it is such a great, yeah, like, just, like, whether wherever you are, whether you're in nature or, like you said, in science, like, just doing something where the culture is, yeah, better than where you left it, and I do think that it's changing, I think that there's a lot of things unsustainable about the way science is. I mean, science is obviously, obviously, yeah, obviously I love science and I love it as a means of finding out things about the world, but, like, it's not in a vacuum and we need so many other things, like, even just if we imagine, like, what's done in the lab with looking at patient cells, I'm sure a lot of that is, um, dependent upon people that are out there in the communities, like, caring for patients and encouraging people to participate in science and, a lot of the community engagement that drives the science and then, right, vice versa. So it's not just, yeah, scientists can't just really sit in the lab and not talk to anyone. I mean, maybe in the past they have, but I think that time is kind of over where, like, that's a way to do it. I don't know.
Yeah, I agree. Like scientific outreach these days and scientific communication has so like it's such a broad umbrella now and it makes me so excited to see because like I follow so many different scientific creators and some people are just like making jokes all day and that's one form of like
Right.
scientific communication that I like. Other people are like telling you about their niche science, which is really cool, and other people are like, hey, this is a PhD journey, I want to take you
Right.
on this. And then there's like me, I just am somewhere in between, I don't know, but like there's so many ways to do outreach, at least on social media, but then there's a explosive amount of outreach that's happening even more now, where even just leaving my university, I see people protesting so much, like, to protect science, to protect the CDC, and, like, outreach is a big part of science, and I think we're realizing it even more now, which is exciting. And the part that I was talking about that I dislike is, like, we still do have like that old kind of mindset, and some people that are like still gripping onto that kind of like older way of being. And I'm like, you're gonna come around one day, and if you don't, you're gonna get left behind, which is so sad to me. But
Right.
yeah, it's an exciting time to be in science, even though it's scary. Like
You're
it's
right.
terrifying, but it's exciting too.
Yeah, well, I mean, we're on the cusp of so many things. I mean, some of the same technologies that are really scary for me, like AI or, I mean, robotics, right? Like anything. I mean, there can be scary and negative aspects of those things and kind of for me communication, like I have a very much of a increasing hate for anything like AI generated content, right? But then when we're talking about the robots that are taking care of the organoids or AI to be able to just to like break apart more complex science and really understand it or large scale like data analytics and being able to understand human diseases better because we can like ingest and process more data. Like those are wonderful aspects of it, right? So it's just like deciding like what do we want to use using power for good versus evil, I
Yes.
guess. I don't
Yeah, 100%. Like, even with AI, I feel like when I learned about it years ago when I was in my post-bac, um, it was a tool that was created for scientists by scientists, and there was, like, this idea where we could take all of this genetic data that we're generating that is, like, seems impossible for a human to comb through and find something meaningful. We would be able to use this, like, maybe find hits across certain diseases that we could then go investigate. Um and now that it's, like, so open access and, like, it's being used for so many different things, it is kind of terrifying to see. Not even kind of, like, it is.
Yeah,
And the global impact that it's having is also scary. But like you said, like, these are tools, and we can use them for good if we so choose. Like, even organoids are scary to people, and I would agree, like, at some point, we're gonna have to put some ethics on this thing and say, like, at what point are we getting so close to human that we need guardrails for these types
right,
of experiments?
right,
So, yeah, like, knowing the tools, but also having a foresight to know how they could be used for bad, and then putting rules in place is important for every field, I would say.
right. Yeah, it's so true. Well, I'm gonna circle back just before we wrap up because I remember now a question I was gonna ask you at the beginning, which was like which is maybe kinda deep, but I think it's very interesting, is that, like, in dementia or Alzheimer's or ALS, a very complex disease like some of these things, you had mentioned that a lot of cases of these diseases are, like, somatic, meaning, like, we don't necessarily know what caused them. There's probably, like, right, a variety of different genetic mutations kinda coming together that create them as opposed to, like, one rare genetic mutation that if you have it, you're pretty much gonna get this disease or like it's very likely. What I thought was interesting at NIH CARD is that like it's really difficult to study the disease and how it's progressing and what's causing it if you're just looking at these somatic cases because it's like because you don't know, versus if you look at some of the rare yes, maybe it's rare that this one genetic mutation causes this, but by studying that one in particular, you can tell, like, okay, what first starts the disease and then, like, what happens next and what happens next, and you can kind of find more of the process where then you can take that and say, like, "Okay, now can we find hints of that back in the, like, more general cases or the more somatic cases?" Is that true? Do I have that this idea right that, like, maybe some people might think, "Oh, well, if you're only studying the rare case of it, maybe that doesn't help everybody." But I mean, by understanding the biology of that case, it's maybe a little bit easier to study. You can learn more that then you can like generalize or apply to other, maybe more complicated spectrums of that disease.
Yeah, I think you 100% have it correct. Like, and what you're talking about is something I was so pigheaded about when I was a younger scientist because I was like, I don't want to study this one specific thing. I want to
Right.
study like these big things. But now that I'm here, I'm like, wait a minute. So like what you're talking about is like a lot of diseases are what we call polygenic. There are a bunch of genes contributing to this one disease a lot of times. There are very few cases of things that have one known genetic mutation and we can correct that and then that person is fixed. But
Right.
even in trying to uncover what that one gene is, we found out so much because one gene could be touching so many different processes in the body. And trying to understand all those processes is that's usually an entire field. Um, and so with these sporadic cases where we have no clue, what we do know is, at least in ALS, is that it's still ALS. Like you still have this motor neuron degeneration and you still see these patients, losing their lives sadly over this short period of time. So there's like this commonality. And like you said, by studying these known genetic mutations, hopefully we can find some cure, some treatment for these patients. But what I found with what I think the field is finding with ALS is that even though they have this genetic mutation, it's kind of seeming like it might be polygenic where, yeah, this mutation is present, but like there's still the environment and all these other genes that are acting upon this. And we still don't know what that is because experiments have been done to correct these mutations and it doesn't cure the
Oh,
disease. Yeah,
gotcha. So, yeah, so it's still probably interaction of that, even if it's one genetic mutation, interaction with lots of things like lifestyle, environment, and all kinds of stuff. Yeah, it's just so complicated. You realize it doesn't take long before, as you study more of these things, it's like, oh, why don't they have a cure for cancer yet? Or this one, then you realize like how complicated these things are. There's like it's not just one fix, right? It's like so complicated. There's so many things you need to target. If sometimes you just look at like the symptoms at the end and you treat those where you're not treating the very like beginning of the cause.
100%. Like, and that's the scary thing, especially in ALS, is like we don't know when onset is. Like, not truly. Like, we know when symptoms start, but we don't know when the disease starts. Which makes organoids a cool model because they're more neurodevelopmental, so they're younger. People conceptualize them as being younger, so you can see these differences early on, not so much in the later stages where it seems like there's not much you can do outside of just, like, making sure the patient is comfortable and trying to, like, slow the disease with various drugs. So, yeah, I I all that to say I agree.
Yeah, no, that's awesome. Super cool. Thank you so much for chatting and showing the lab today. I really appreciate it. And I don't know, did you have anything else that I didn't ask about that you wanted to share?
Not that I know of. I've had a blast just, like,
Yeah.
chatting and, like, getting to talk about science outside of talking about it with people in the lab, so it's been
Yeah,
really
exactly.
fun.
Yeah,
Thank you for
I
having
love
me.
just connecting different areas of science. I mean, like, oh, that's like this thing over there. And, because there's so many things I think we can learn from each other too, of like, yeah, someone studying ants makes a discovery, and you're like, well, actually, I'm going to look at that for neurons.
I mean, that's how we make discoveries, like,
Yeah.
studying all the different things, and then sometimes those discoveries can be used across so many fields, like, cool stuff.
Yeah.
Really cool. Yeah.
Awesome. Well, thank you so much, Nuri.
Thank you.